Hello, everybody. It looks like I might have to go at the speed of a Japanese bullet train to complete this. So, yes. So I'm a PhD student, and my just recently entered my second year, and we've been studying invertebrate venoms. So this is one of the scorpion venoms that we have tested from an array of 60 venoms. So that is why I'm going to show one of the venoms and their biochemical and functional characterization. So there are like over 2,000 species of scorpions, and they possess various aspects or properties. Like most of it is neurotoxic towards its prey base. And like many of the studies on scorpion venom has focused on the toxins, exploring the neurotoxic aspects of scorpion venoms. So what we are doing differently is that in our lab, we focus on studying cardiovascular diseases, especially thrombosis and effects on clotting disorders. So we wanted to test the potential effect of invertebrate venoms against any effect on the clotting processes as well as vascular system. So that is the one of the reasons for choosing this study. So this is the particular species that out of the array of 60 venoms that we initially identified as potentially showing unique activity in terms of enzyme activity, and hence we have chosen this. So this particular scorpion species is from North America, especially from the border of Mexico and the state of California in USA. That's its in a native range, and this has a very similar sister species called Paravejovis spinigeris, which is a little bit more known as a devil scorpion. So these scorpions are having venom compositions which are unexplored as to my knowledge, and therefore we also want to know what is the venom composition, because these might offer some unique properties in terms of its effect on our clotting processes. So the main hypothesis or the novel is that these venoms could have any effects, because we do not know what is inside the venoms. We do not know whether they have any applications in terms of therapeutic potential. So a prerequisite is to characterize them initially in terms of biochemical properties as well as other effects in terms of circular clotting process. So some of the techniques that I've used in my project are first to establish a biochemical profile in terms of three major enzymes, metalloproteases, serine proteases, and phospholipase A2. And subsequent techniques involved studying the effect of the venom on platelets using flow cytometer-based assays and aggregation-based assays. So in terms of enzymatic activity that this venom has exhibited, it appears that metalloprotease and serine protease, it does not possess metalloprotease and serine protease activity. However, its PLA2 activity, it's a short form of phospholipase A2, appears to be very significant. As you can see, even at a very small concentration of 3.1 micrograms per ml, it is exhibiting significant phospholipase activity. So again, since the venom's composition is unknown, we do not know whether it's what structure it is or what its role could be. So this is an initial screen to find out establish its enzymatic profile. So before I go to the other techniques that I've used, just a brief overview about why we're using the platelets. So platelets as one of the main substrates for the venoms to react with. So we are studying the primary hemostasis aspect of clotting, where after injury occurs on a blood vessel or a vascular site, platelets are free floating in the blood. The collagen is exposed upon the cut for the endothelial layer, and these attract platelets to bind to them. As you can see in the number 1, that's marked as platelet adhesion. Upon binding with some of the layers or the collagen, like you have shape changes of the platelets that occurs. So this exposes certain elements of the platelets to the surface of the platelet cell membrane, one of which is called P-selectin. As you can see in the picture on the right, P-selectin in a resting platelet, that is a free-floating platelet that has not yet been activated, is inside the cell. And the GP2B and GP3A are receptors that are receptors for fibrinogen, which is also a free-floating molecule in the blood vessel. So why these two are important is because for the technique that I use, flow cytometer, the flow cytometer technique that I use, we are studying the effect of the venom on these two markers, so fibrinogen binding levels and p-selectin exposure levels. So once a platelet moves from the resting position and gets activated, which is signifying start of a healing process, the receptor that you can see there, p-selectin is exposed on the surface of the platelet whose shape has been changed, as well as the GP2B and GP3A form a complex that helps to bind with fibrinogen. So fibrinogen binding helps other platelets to be bound to it, and a platelet plug is formed. So that's how the clotting process roughly works. So when we tested this venom against platelets in a flow cytometer based assay, we discovered that in two types of you can see there it's written in the graph venom without agonist and venom with agonist. These were the two primary modes of experimentation we did. So the first one refers to treating the platelets with venom alone and seeing if its effect by itself. And the second column represents an experiment where we have allowed the venom and the platelets to interact with each other for a defined time period, after which an agonist was added. So here the agonist is ADP, adenosine diphosphate, and which is a well-known inducer of platelet activation. So this has appeared to inhibit the agonist-induced platelet activation very significantly, as you can see. So the two markers, as I said before, are fibrinogen binding and p-selectin. So these are the two markers for platelet activation. So it has inhibited both for fibrinogen as well as p-selectin exposure in terms of inhibiting the ADP's action on platelet activation. So interestingly, this same effect has been observed in another invertebrate venom that we tested on platelets, which is the sister species of Paravajovis confusus, as you can see on the two graphs to the right. So that also has exhibited similar properties in terms of inhibiting platelet activation. So one of the other techniques that we used was to, in terms of studying primary hemostasis, is aggregation assay. So here, this is a representative trace of platelets aggregating after the initial activation has occurred. So in the lines represent different, so the first line is saying positive control, which is again the agonist that we have used, which is ADP. So how this trace works is that if you can see the first minute on the x-axis, that denotes the time when the venom and the platelets have begun to interact with each other. So first we are testing just the interaction of the venoms and platelets alone. So that has happened until 5.5 minutes, after which the agonist, which is the ADP here, has been added. And then we are seeing if platelets, if the agonist added has been influenced by the venom. So as you can see here, so the red line represents the positive control, which is giving around 70% of aggregation, which is expected because it's a known activator as well as aggregator of platelets. So but with the venom, there appears to be a dose-dependent manner in which it inhibits the aggregation of platelets. So the higher concentrations of 50 micrograms per ml and 25 micrograms per ml appear to inhibit the aggregation of platelets. Whereas as the concentration gets lower, the venom appears to act in a weak manner and the ADP appears to be able to aggregate the platelets. So this is another demonstration of how this venom has affected platelets. And apart from this, we also have tried to get a protein pattern about what this venom might hold as a preliminary data, of course. So from this SDS page that electrophoresis that we did, you can see that there's a predominance of low molecular weight proteins. These could also be peptides. And there are a few components that may be at 50 kilodaltons and at 37 and 25. So likely that since we do not know maybe like PLA2 enzymes in scorpions, since they are well known in snake venom, we do not know in scorpion venom what kind of structure they would have or what sizes they might possess. So we do not yet know what these abundance of low molecular weight proteins might consist of. So we plan to do further experiments to characterize these other peptides and the venom composition. We aim to purify some of these proteins through chromatography techniques, as well as then we can go ahead for mass spectroscopy. So this is the basic characterization in terms of creating a biochemical profile for this venom, and then like studying its effect on platelet cells. This would have so to see what kind of effect it would have on the platelets function. So it's a short presentation. And I'd like to thank my supervisors, Professor Sakti Vayapuri and Professor Ketan Patel, and Dr. Steve Trim, collaborator, who has generous provision of 6 array of venoms for us to test, which is once again lyophilized in crude venom form. So thank you very much.
Thank you for ending that session for us. And are there any final questions for today?
Hi, I'm Ornella Rossetto from University of Padua in Italy. Thank you for your very nice presentation. Do you think that the inhibition of aggregation and activation of platelet could be, could derive from the PLA2 activity of the venoms, maybe for the formation or release of
Yes.
phospholipids and fatty acid, and so damage of the membrane? Could you possibly inhibit the phospholipase activity, or try to,
Yeah, thank you for the question. So I do not know, we do not know what is the mechanism by which this venom has acted. So the venom used was a crude venom, which is consisting of a mixture of venoms. So we have screened for 3 specific venoms, enzymes, which is metalloprotease, styrene protease, and phospholipase, because we think they might have an effect on blood coagulation, as has been seen in snake venom. But we are dealing with invertebrate venoms. So it could be having an effect on primary hemostasis. That is like PLA2, it might have. But again, we will have to find out like what is the exact mechanism, if at all it acts on that. So it will be one of the objectives that we'll be doing it in future. So thank you for that. So.
Any final question? You're off the hook.
Okay.
Thank you
Thank
so
very
much.